Little p, and please write this in, because understanding these little differences will really help you. This p and this little q represent the frequency of all of the population's dominant alleles and all the population's recessive. It's really the probability, right, when you think about it. Okay, and I want you to understand that little p really represents, hey, how many t's are out there? And little q represents, in the entire population, how many little t's are there, right? We're not counting individuals. We talked about hemophiliacs who are homozygous recessive for that two broken alleles for the blood clotting factor. Okay, those individuals. We want the individual, okay, frequency. And again, a frequency value, not a percentage, it's out of 1, okay? So we found, okay, the individuals that are what? Homozygous recessive to be 0.3. 3 out of 10 was basically what we found, okay? But that's not what this is. This is Q, all right? So it's very important, okay, you understand that difference. So the frequency of the P's in society and the frequency of the Q's in society, okay, have to equal 1 for the development of the gamete, the haploid cell. What? With haploids are either going to get a P or a Q. So these are all the possibilities. These guys are mutually exclusive to each other. So they both have to equal 1. Okay? Now, when we mate this with another gamete who happened independently of making this one from a female gamete, they have the same scenario, that the total what? Combinations of P's and Q's will have to equal 1. Because making the female gamete, you're either going to get a P or a Q in that gamete. It's one or the other, so the possibility is still 1. And because we're now going to mate the male and the female, okay, we add all the possibilities that this single chromosome could have for this allele, and this has the same scenario because there's only P's and Q's. And because the making of this gamete is independent of this one, we multiply, right? Because the meiosis that occurred for this male in spermogenesis is different from oogenesis, the egg that was made. And so we multiply these. And so when we do that, I know some people do it differently. I used FOIL, okay, when I was growing up. I don't know what the New York State math is now. But I'm going to do first, right? Binomial, P times P, so the first. So that's going to give me P squared. Okay, then I do outer, P times Q. These are my outer ones. So that gives me plus P and Q. And then I do my inner. Again, P times Q is again PQ. And then I do last, Q times Q, and that's Q squared. And if I simplify, I get P squared plus 2PQ plus Q squared. Now this equation equals 1, and this is so important. Okay, so I'm going to change colors. Okay, this is for individuals. We can use this now. This is the Hardy-Weinberg equation for individuals in a population if there's no what? Small population, no selective mating, if there's no mutation, okay, no influx of gene flow, and no selective pressure. Okay, this is how you figure it out. So we can figure out exactly how many homozygous, because right now it's P and P. Don't forget, the little p represents the dominant allele. So let's put that back in there. So we don't want to lose sight to what this all means. So yeah, we'll stay with red. So this guy really is TT right here. That's what P squared is. Remember, the P represents the dominant allele. Who is 2PQ? That means you just have 2 times, okay, 2 times what? Big T, little t. And Q squared, 2 Q's, remember the Q's represent the what? The recessive allele is the TT individuals. Don't forget the 2 is there to times it by 2 to make it even, but party people, okay, there are your 3 possible what? Genotypes, okay? Either the homozygous recessive, the heterozygote or the homozygous dominant. There it is. Okay? And so, again, I don't care, Mr. Kratz, what's the big deal? Well, I can see if, in fact, we're in Hardy-Weinberg equilibrium, if our class data reflects the data that we have in society. We kind of mentioned that 30% of society is non-tasters, which means 0.3 is my frequency. Now, from my leaping, which one of these do I definitely know? And this is important. My leaping off or my point that is we always take off from is from homozygous recessive. Now, if you look closely of my math here, that gives us the equation to figure out the frequency of these types of genotypes, okay, using this Hardy-Weinberg formula. Notice there's what? A 1 to 2 to 1 ratio of those types of individuals. Notice if we would do a simple Punnett square of a paternal and maternal, okay, you'd get the what? 1 to 2 to 1. And notice this is the leaping off, the jumping off point. And the reason I say that is because if I know this guy right here, we know Q squared. We found Q squared. What was Q squared? Go back to the original. Go with me. Go back. Flip this page over. Okay, let's go back to this page. Please flip that page. This, my friends, was Q squared. I should write with it something I can write. Here we go. This is Q squared, the non-tasters. So we have, okay, I'm going to change this up. Yeah. We have the Q squared frequency right here. That's what we can get by doing this activity. We found the frequency of my Q squared or my homozygous recessive individuals. We have that. And all I have to do is solve for the individual P and Q and fit them into the formula to figure out my other genotypes. I cannot figure these guys out because the tasters were either or both of these. I can't discern between these. So our jumping off point is the homozygous recessives. They're the one that are expressing a phenotype, okay, that's so different from the others, and they only have one genotype. I can't say that enough. You're going to keep your life so much easier if you understand that this is your Q squared, your homozygous recessive. That's going to make your life easy. So now that I know that this is Q squared, and I know there's a formula for the rest, okay, I can do this. All right, now, let's go back to that page. Turn back. Let's go after what P and Q is. All right? I want the individual P and Q. Again, these would be the frequency of the individuals in society. Notice they have to equal 1, right? These are going to, all these numbers, all these probabilities to be these individuals have to equal 1. These are all the what? 3 probabilities, or the 3 types of individuals, okay? So if I had a 3-sided die, which is kind of weird to be a triangle, I guess, I'm not sure. But the point is, if I rolled it, I either get a 1, 2, or 3. Okay, so it has to equal 1, the addition rule. So we're going to go after the individual allele frequencies first, to go back and solve for what the frequencies of my homozygous dominant or my heterozygotes, okay, I need to solve for my P and Q. And the jumping off point, I know I'm being redundant and tall, is my Q squared. So what I'm going to do is, I'm going to go find Q. And like I said down here, to get Q, I take my Q squared, and again, I know that my what? I'll say it again. The TT individuals, the non-tasters, okay, from the first page is 0.3. But that is really what? Q squared, right? Q times Q. All right? So, if my Q squared is equal to 0.3, how do I get this to equal just Q? Well, of course, I'm going to square root both sides, right? So I just square root my Q squared. Okay, as I put over here, I take my square root of my Q squared to get Q. So let's do that. All right, so square root of 0.3 is going to equal my Q. Remember, 0.3 is equal to Q squared. So to get Q, you square root both sides. So take my calculator, and I'll do square root second function of 0.3, close my parentheses, and I get, okay, Q is equal to 0.5477. Okay, that is what the frequency of all of the Qs in the population. Not individuals, all the chromosomes that are in living organisms that are carrying the Q or recessive or broken allele. What's the other possibility? Be carrying the what? The dominant, the P. And what do we know? Changing my ink, because it can do it. Okay, what do I know? Well, I know P plus Q has to equal 1. Why? Because that's all the possibilities. So if I know my Q is 0.5477, right? How do I get P? Well, subtract from 1. 0.5477. I should do 1 minus that. Okay. And I get 0.45, say 23. And if I was to, what is this equal to? This is equal to my P. And I may have rounded differently. I think I'm okay. If I add these together, it should give me 1. So all I did was take my Q, which I got from my homozygous recessive individuals that are Q squared. So to find Q, I square root to find the frequency of the recessive alleles. And I subtracted from 1 because x plus this equals 1. So solve for it algebraically. You're going to subtract that. You got the P, because this plus this equals 1. And now I can do this math. I have my P's and my Q's. And now I can go and solve and plug back into the formula. So let's go do that on the next page. So I guess I should have rewrote that. All right, so I got for my Q to be 0.5477. And then I got my P. You can see the P was 0.4523. Okay, and those are my frequencies. I left a lot of numbers there just to not mess up my rounding. Of course, these are the frequencies of the individual alleles in society, and these would represent the ones that are not changing if these 5 things we talked about are held true. Okay, so let's take that now, and now that we have this formula, okay, let's go solve for the rest of this. So I guess I'm rewriting stuff all over the place. Okay, so P is 0.4523, 0.4523, and my Q is 0.5477. And why do I have that in green? I want to keep telling you we get this one first, because this came from what? What did Q come from? If you understand this, boy, you are really, or girl, you really are on to something. It came from my Q squared. What is Q squared? Oh, that's Q and Q. Isn't Q recessive? Oh, that's T and T. That's individuals. So by counting our individuals that are what? Inflicted. People that don't taste the bitterness, we're going to call you inflicted, sorry. Okay, only because I'm a taster. Sorry, but in any case, we can see in society, who is recessive by noticing that they don't have a working gene. So there's some issue, unfortunately, possibly a disease. Now, being a non-taster is not a disease, but hey, I'm a taster. But the point is, I'm the cure. The point is this, T and T. We can see this. In fact, let me just draw an eyeball. So we always start, that's a crazy eyeball, I'm looking, see these guys and count them. We weren't able to count who was homozygous recessive. We weren't able to count who was homozygous, who was a heterozygote. We can't count those genotypes. Okay, it's important that we can't do that. Started off, okay, with this individual, the Q squared. So who's my P squared? What is the frequency of individuals that are homozygous dominant? Don't lose sight what this means. Remember, P is what? Big T. Q is little t. So P squared is big homozygous recessive. These are my heterozygotes. And this, of course, we just found out from our data. Our data gave this homozygous recessive. Okay, so let's go find P squared. All right, so wherever I do with my calculator. So I already have my P. Don't forget how we got P. We always get our Q squared first. We did the square root to get this, and we subtracted from 1. And now we're going to do P squared. So I'm just going to take my 0.4523, and I'm going to times that by itself. It would have been easier if I just hit the x squared button, but I didn't. So I get 0.20, don't care about rounding here, 0.2046, we'll just round to 4, we'll just keep it like that. And then my 2 PQ individuals, notice the 2, so I'm going to take 2 times, my P is 0.4523, and it'd be great if I wrote it that way. All right, I can do this. 0.4523, and I'm timesing it by the what? The Q, which is 0.5477. I definitely did not give myself enough room, so all I'm doing is 2 times this times this. Let's put that in there. Put this in my calculator. So 2 times 0.4523 times 0.5477. And if I'm not mistaken, and there's nobody here to correct me because, boy, I'm going solo now, the frequency that I get, okay, let's keep that, whatever color that is, 0.495, let's go 7. Okay? And so, if you do a eyeball, shouldn't all the individuals equal 1? Right? Isn't that formula right there? So shouldn't all this equal 1? Absolutely, right? So don't do the individual alleles, do the P squared, so 20 plus 50, 70, 70 plus 30, it's 100. That makes sense. These are all the frequencies of the individuals here. Okay? And that, of course, is the I guess I should have put the calculations above and the numbers below. So they're my frequencies here. All right, so I guess I didn't follow my own worksheet. Okay? Now, interesting enough, how many people are in the class? Well, we know 0.30 because that was our jumping off point. 30% of the class were non-tasters. They were the homozygous recessive allele. But how many people in the class, based on Hardy-Weinberg and based upon no evolution, do we have? Well, take your 0.4957 frequency, times it by the 20 students we had in the class. Right? Because if you look back on our original work, okay, we had 20 students, right? It was out of 20 that we did our work from, right? So, back to here. And so, you take this frequency and times it, so 0.49 times that by 20. And I get 9.9 students, which is going to round off to 10. Okay, because you can't have 0.9 of a student. So I got 10 students. Okay, this will not be 0.3, 30% of my students, right? So 0.3, I can do this, 0.3 times my 20, right? If you want to do that. That was my 6, right? And then, of course, do the math. Okay, what's 20% of 20? So 0.20, so this one is 0.2046 times the 20 students. That's how I'm getting the total number of students. Out of the total number, remember, this is the frequency times the total number of students gives me the actual number. That's what we're doing here. So 0.2046 times 20, and I get 4.092, which rounds to 4. And lo and behold, 6 plus 10 plus 6, of course, that gets me a total of 20. So what Hardy-Weinberg gives us is, although we know already 6 out of the 20, the 30% were non-tasters, it's actually giving me the percentage of what the other genotypes must be if we're at no, what? No evolution going on. That's pretty cool. It was 14 total, but only 4. Most of them are heterozygotes. Look at that. Is there a heterozygote advantage that still persists in our, what? In our population? Maybe. Now if you go to the last page, okay, go to the last page, and it's really interesting to see what the frequencies of the PTC tasters are. Okay, look at Australian Aborigines. These people are the ones that have the least amount of gene flow. They've been together as a group longer than other people, so they have a higher non-taster part than I'm sorry. The Australian Aborigines have a huge non-tasters. Look at the kind of the error bars here. They're not really error bars, they're the mid-range, the percentiles. So this could be, if you think about this, the 25th and 75th percentile. If these are medians, these would be the boxes, right? So these could be box plots. Very interesting. Notice how small that box would be. There's almost no variability. All of them are 49.7. They have so few gene pool, okay, they don't have any genes mixing in, so they're about 50% are non-tasters. What does that mean? Well, we're not really sure, but that's a selective advantage, I guess, okay? Whereas the other people have much lower, okay? And look who has the lowest, Central America, or the Caribbean, okay? And again, you could try to make some sense out of that, all right? And again, if you look at this, early studies show that the ability to taste or related chemical was under genetic control is probably a simple Mendelian trait. People have argued that there's more complexity. Why this trait is maintained in human populations is unknown, but several studies have shown this polymorphism. Okay, remember, polymorphism is basically a change in the DNA called a mutation, which is a non-tasters, right? May influence food selection, nutritional status. Not really sure to make of this, but it's interesting that the Aborigines of Australia, okay, have the greatest percent of non-tasters, okay? And so they don't taste bitter. And maybe that gives them an advantage to eat food that's better for them. But does that give them a disadvantage against the poisons? Maybe, okay? They, of course, live more in a less modern society, and so they have probably a selective pressure, making them have a greater amount of non-tasters. We don't live under that same, or most of these people here live in a modern society, so therefore the selective pressure exists here. Okay, and you can stop and think about what that may be, but my guess is being non-taster is there's a selective pressure here to keep them this high. All right, and then of course no gene flow there. And what that may be, okay, my guess would be that it allows them to eat food, since they don't get food shipped in like we do, get kiwis and bananas from different places around the world. They eat what's in front of them, and that allows them to eat foods that make them, even if they're bitter, still good for them, and therefore survive better by not having that bitter gene. That's my guess. Hope this helped.